incredibly interesting paper came out recently that showed something unprecedented for the very first time. It showed that somebody's cancer sample had spike protein in it. The reason why this particular publication made some controversy is also based on the fact that this was an individual who was mRNA vaccinated, and based on some of the work that the authors of this publication did, they came to conclusion that it might have been the vaccine that caused the presence of the spike protein in that cancer sample. However, I don't necessarily agree with this information. I will explain to you why the way that information has been presented might not necessarily be accurate, and we would need better evidence to come to that conclusion. But let's break it all down. My name is Dr. Mikola Rashik of Neurogenomics, and let's get started. So this was an 85-year-old person who presented with a skin lesion and went to a doctor, and the doctor assessed it, and it turned out to be cancer. And this was about a month after this individual had their 6th mRNA shot. All right, so you have that temporal association between the shot and this lesion. And let's take a look at the pathological examination. We have this figure from one of the blogs, because the actual paper is behind a paywall. And you can see on the left-hand side of this figure, there is that skin lesion. But let's take a look at on the right-hand side of the figure, and that's the biopsy, skin biopsy that you see. And you see a bunch of different colors and that are cells stained with different colors. Now, what that is hematoxylin and eosin staining. I hope I got the name right. It's a very standard name, typically just abbreviated as H and E. Now, hematoxylin stains nuclei, so dark color, like dark purple color nuclei. Now, eosin instead stains the cytoplasm, the liquid component of the cell, as well as collagen. And you see at the very top of that image, you see white space. Basically, there's nothing there, and then immediately you see this pink layer. That's the top of the skin, literally your outside of the skin that faces the environment. And below that, you see this darker stained pattern, this purplish wavy pattern, and that's your epidermis. The reason why it stains a little darker is because it has densely populated cells, and therefore there's more nuclei to stain, and as a consequence you have this darker pattern. And below you have a lighter color skin pattern, and that's dermis. So that's the reason why it stains lighter is because there's fewer cells and there's more connective tissue there instead, collagen, and therefore it stains a little lighter. Now, within that image, you also have cancer cells, and they labeled it. They labeled the first ones we're going to focus on are invasive ductal carcinoma cells. And these are pointed out with just regular arrows. Now, those are the cells from this individual's past breast cancer. Now, invasive ductal carcinoma is the most common type of breast cancer. So ductal means it originates in the milk duct, and then invasive, of course, it means it can spread to breast tissue, other parts of the breast tissue, and carcinoma, cancer. And that's what you would expect. But also with the arrowheads, they point out to additional cancer cells, what they refer to as epidermotropic cancer cells, ECC. I hope I got the name right again. What's unusual about that is that it means this cancer is invading higher or upper layers of the skin, which is why this individual presented with this skin lesions that were quite obvious. But what's unusual about it is that these cancer cells, they're invading upper layer of the skin as opposed to breast tissue. So it means it's fairly aggressive. Typically that would indicate that the prognosis, it's a worse prognosis for such an individual. All right, so that's why do I tell you all of this, and why did I even show you this figure, is because the authors of that paper decided to investigate both of these type of cells for presence of the spike protein, which is the first that I'm aware of. As far as I understand, this hasn't been done by anybody else yet. And that brings us to the second figure. And in that figure, what you see is that let's look at the very bottom. Those are the original cancer cells, the invasive ductal carcinoma cells from the past. And you can see that there is no spike protein there. And I investigated for two types of proteins, spike protein and nucleocapsid protein. And then they investigated the metastatic invasive carcinoma, ductal carcinoma cells, after it came back in that skin lesion, as well as that epidermotropic cancer cells. And both of them present spike protein. You can see these brown points and arrows point to it. Versus nucleocapsid protein, you don't see it at all. Now, what does that all mean? First of all, let me tell you what they did. They looked for these proteins via a technique called immunohistochemistry. You target the protein of interest with an antibody that is attached to a certain complex. I'm just going to call it complex for sake of simplicity, that allows a chemical reaction to take place, and that's how you see this brown color. So this allows you to target and find protein you're interested in with a color. So it's colorimetric assay, and that's how they were able to spot the spike protein, but not the nucleocapsid protein. Now, why is that significant according to the authors? Is because nucleocapsid protein is a virus, SARS-CoV-2 virus protein, as is obviously spike protein, but spike protein is on the outside of the virus, attached to the membrane of the virus, but pointing to the outside world, versus nucleocapsid protein is found inside the virus where it interacts with the genetic material of the virus. By looking whether there is nucleocapsid protein present or not, and discovering that there is no nucleocapsid protein, authors come to the conclusion that the spike protein that they observed must be from the vaccine. However, I'm saying no, that's a wrong conclusion, and unfortunately very commonly made mistake in published scientific literature, where there's an assumption made that just because you don't see nucleocapsid protein from the virus, it means that the spike protein in vaccinated individual had to come from the vaccine. But the reason why I see that wrong is for a simple fact that spike protein might simply not be degraded as fast as nucleocapsid protein, and therefore a person could have been infected asymptomatically, even no symptoms, they could have been infected, nucleocapsid could have been destroyed faster, and spike protein persisted longer, and that's how you see only spike protein, not nucleocapsid. And we know this, for example, from long COVID patients, people who were getting infected, and it was seen that spike protein lasts longer than nucleocapsid protein, and it's known that spike protein can last long times. However, as far as I know, there's only one way only that you can demonstrate presence of spike protein from the vaccine, mRNA vaccine or any vaccine, and that is by investigating its amino acid sequence. So remember, proteins are made of amino acids. Genetic material is the blueprint that tells the cell how to make a protein, and the protein will read genetic material to start linking amino acids, the building blocks of proteins, together into a contiguous chain. And because amino acids have electrostatic interactions, they start creating three-dimensional shapes and form a little robot that will do some function inside your cell. Right? Now, vaccinal, for the vast majority of vaccinal spike proteins out there, mutations were introduced that are not present in the natural viral protein. And the reason why this was done is because spike protein can, once you rip off its head, it has these tentacles that it can sprout out, and these tentacles can grab a cell and pull a virus towards the cell so that the virus could fuse with the cell. And mutations were introduced, these specific mutations, proline amino acids were introduced, which are really weird, bulky amino acids. They're not very flexible, and by putting those prolines, two prolines, in a specific spot of vaccinal spike protein, it prevented those tentacles from being mobile, which means that if you were vaccinated with spike protein construct, the spike protein you would have produced on the surface of your cell would not be able to spread out those tentacles, grab another cell, and make two cells fuse themselves together, which happens with the virus. This is called syncytia, and it's a signature of pathology that we saw with COVID-19. Now, we did not want to have that with vaccinal spike protein, so those prolines were introduced. And the only way for you to find out whether spike protein is from vaccine, you have to inspect that amino acid sequence. And as far as I know, there is only one scientist that has ever done that. And this was spearheaded by a team of Dr. Carlo Brogna. And this is, as far as I know, the only study that has investigated this and shown that spike protein from vaccine can persist in blood for up to 6 months. That's only as long as that study was conducted, so we don't know whether it could last even longer or not. It's just simply the longest time duration that team of individuals have investigated this, and I did a video on this. Now, everyone else who claims presence of vaccinal spike protein, they never actually double checked it, they never confirmed it, so it's just a guess. An example, not long ago, a study came out from Yale that investigated post-vaccination syndrome and to what degree spike protein from vaccines might be persisting in such individuals. And they made a famous claim that even more than 700 days spike protein could be persisting in such individuals. But they did not check the amino acid sequence, so you cannot recount the possibility that such spike protein simply came from asymptomatic infection in such individuals. And having asymptomatic infection is it's not impossible, quite the opposite, it's very possible. So that's the only way to check for the presence of spike protein. And these people in this cancer sample, they did not do it. And as a consequence, this does not confirm this. Now, I argued this point amongst a group of scientists. I recently was invited to participate in this article, which, by the way, is now available for as a preprint. So we're waiting for a publication of this. And I argued that point, and it actually changed the structure of the article. Now, by the way, Dr. Carlo Brogna is one of the co-authors of that article. I really admire his work, so it's very cool for me to be a co-author on any work he's participating on. And by the way, that article is on how science is not a concept where information is added to our mutual pool of knowledge and we progress. Rather, science is controlled or can be controlled, and the way science works is that we create paradigms, or the stakeholders can create paradigms and then defend those paradigms at all costs from new incoming information, and it can actually stifle scientific progress. Now, this concept is not new. It's been discussed many times in the past. We just simply reiterated it again, and we used an example of such an immunohistochemistry as I just described to you to describe how science can be controlled by certain entities like this. So, fun article, check it out, available on preprint. So take a look. And I got into deep conversation with one of the scientists. It's a great privilege for me to work with these people because they are very smart, way smarter than me. I'm like smart, but I'm smart like a average street person smart, not like brilliant smart. These guys are smart. The way they can connect the dots is fantastic. It's really fun to be able to be exposed to this kind of intelligence and learn from these guys. Now, so that's why I argue also against this too quick of a conclusion that this might have been from vaccine. Now, of course, there is that temporal association. This person had 6 mRNA vaccines as well, 1 month after this cancer was presented. But then where did it come from? Where did that spike in the cancer sample come from? So there's few possibilities. So obviously we just discussed the possibility of asymptomatic infection. Next possibility is that it might have come from DNA contamination that we now know is present inside mRNA vaccines, both Pfizer and the Moderna. Now, this story was broken to the world, probably more than a year and a half ago by Dr. Kevin McKernan, I believe. I hope I pronounced his last name properly. And now it's been published numerous times that this is true, that there is this DNA contamination inside the mRNA vaccine. So the plasmid that was used to produce the RNA that acts as the gene therapy or vaccine was left over inside these vaccines. It was cut up so that it was fragmented, and as a consequence, it was assumed that this would become benign. But we really don't know, and there is now incoming evidence that some fragments might be quite large, and as a consequence, such a DNA that might have been inserted into a person's cell could have acted as a template to produce spike protein. Now, that same Dr. Kevin McKernan, he also mentioned once on Twitter or X now that he has seen such DNA plasmids inside cancer samples, but this is never published. So for now, that's just a hearsay. We would need to see whether anyone ever discovers this and publishes this. I wouldn't be surprised if eventually we will see something like this. Now that's another option. Another option is that the RNA from the vaccine or that DNA contaminating DNA inside that same vaccine could have inserted itself into our genomes. Now, when vaccines came out, we're all told it's impossible for the RNA to act as a template to introduce genetic material into our genomes, into our own host DNA. That's not actually entirely true. There are mechanisms possible that would allow this. I covered this in one of my videos. They are just so remote in their possibility that for all purposes we can assume that it is impossible, but it is not impossible. You see, it's just highly, highly, highly unlikely. So we just ignore it. But the moment you introduce DNA into the cell, and remember that DNA was trapped inside that lipid nanoparticle, the little fat bubble that housed the mRNA genetic material of the vaccines or the gene therapy, that same lipid nanoparticle had DNA fragments in it. Therefore, you introduce it straight inside the cell. And as a consequence, you dramatically can introduce or elevate that risk that such DNA fragments could be inserted into someone's genome. Now, recently, there was a publication, a preprint that came out claiming just that they found first example of fragment of mRNA vaccine DNA inserted into human genome. However, through discussions, online discussions with Dr. Kevin McKernan, the same guy I was just mentioning, he mentioned that it's very easy to create an artifact that looks like insertion in a process of sequencing. So, and these authors did not double check that in any way, versus if you have insertions, you are also, or what you think are insertions into the genome. You're supposed to be checking it with other technologies precisely because of how easy it is to potentially introduce artifacts like that. So I'm discrediting that. I'm not taking that article seriously because they didn't do due diligence, these authors. So I'm not following that. Now, another possibility is that the spike protein might have come from a reservoir that is inside our body. So that same Dr. Carlo Brogna, the same guy that is, as far as I understand, the only person to ever show how long vaccine spike protein lasts after vaccination, because he investigated the sequence. He's also his group is also the only group that is investigating the possibility that the virus, SARS-CoV-2 virus, could be creating a reservoir inside our bodies, inside our gut, because it can infect our gut bacteria. I've done numerous videos on this topic. I'm very interested in this idea. So, but if that is the case, then the spike protein could have come from the gut reservoir, either as a virus entering circulation or as a spike protein inside exosomes. Exosomes are tiny fragments of cells that can circulate in the body. I covered that in a large series dedicated to spike protein causing abnormal clots. And I discussed how exosomes can circulate spike protein inside our body. So that could be another way how perhaps that spike protein ended up in those cancer samples. Nevertheless, first example, known example where cancer samples were presenting unusually spike protein on, either on the surface or inside. So what's the bigger picture here? Can spike protein be contributing to cancer? Well, we don't know this. There is some small emerging amount of evidence that suggests that perhaps mRNA vaccinated individuals might have elevated risk of cancer. There's some literature, but too little to be able to know for sure. So obviously there are people who are discrediting that information. We need more to know. But we do know as well that IgG4 antibodies are produced after mRNA vaccinations. And we do know that those antibodies can be a potential danger once an individual has cancer. So such antibodies could help cancer development. And we've seen an example of that recently with pancreatic cancers. So there's some evidence suggesting that maybe there could be indeed certain issues related to the spike protein, but we need more information. All right, I'm going to quit right now because it's getting windy. Maybe one more thing that I'll mention is check out my Patreon. Also, I should mention Patreon. I am going to start a new project where I want to build a guide for healthy living, and I would love to do it with help of the community. I am now going to have a regular free live events on my Patreon as well, where you can come and join and contribute your information as to what should be the top elements we should consider to create a guide for very healthy living. I keep mentioning on my channels that one of the most important ways to fight right now what is happening in terms of the consequence of the pandemic via the spike protein is via healthy living. So please check that out as well. And the supporting patrons will get to vote as to what are the top categories that we should consider. All right, I'm gonna say bye right now. I'll see you guys next time.